Now, when you look inside an animal cell, you'll also find a centriole, which is actually inside its centrosome. But centrioles, this is basically the best way to describe a centriole. It's a in animal cells. Centrioles are located in and form part of the centrosome, where they are paired structures lying at right angles to one another. In this context, they are possibly involved in spindle assembly during mitosis. The centrosome is positioned in the cytoplasm outside the nucleus, often near often near it. A single centriole is also to be found on the basal end of the cilia and flagella. In this context, it is called a basal body and is connected with growth and operation of the microtubules in a cilium or flagellum. Right? It's also involved with cell division. Right? So we look at it this way, we see that centrioles present something of an enigma. I'm reading this from a science book now. Centrioles are present in one on animal cells as the basal region of cilia and flagella in animals, and lower plants in cilia and flagella centrioles are called basal bodies, but the two can be considered interconvertible. Okay? Now, when animal cells undergo mitosis, right, they are considered by some to benefit from the presence of centrioles, which appear to control spindle fiber formation. They control the spindle fiber formation, and which later has an effect on chromosome separation. Research, however, has shown that mitosis can take place in animal cells after centrioles have been destroyed. Sometimes this seems to be at the expense of abnormalities in spindle development and subsequent problems with chromosome separation. Recent research also suggests that embryos, for example, of the Drosophila, arrest very early if centriole replication cannot take place. Right? In other words, it depends on the creature as sometimes the centriole has an effect one way or another. Okay? In higher plants, mitosis takes place perfectly satisfactory in microtubules forming spindle fibers, but without the help of centrioles. The function of centrioles, therefore, remains something of a mystery. Now, we're mainly focusing on animal cells, though, okay? Mainly focusing on animal cells. The structure. A centriole is composed of short lengths of microtubules arranged in the forms of an open-ended cylinder, about 500 nacrometers, nacrometers long, and 200 nacrometers in diameter. The microtubules forming the wall of the cylinder are grouped into 9 sets of bundles of 3 microtubules each. In cilia and flagella, where centrioles are at the base of the structure and are called basal bodies, the wall and cavity architecture is slightly different. In addition to cylinder and walls composed of 9 sets of bundles of 3 microtubules, there are walls of 9 sets of 2 bundles. In both types, there is a central matrix from which spokes radiate as in a cartwheel. You heard that right. In animal cells, centrioles usually reside in pairs with cylindrical centrioles at right angles to each other. Centrioles organize a cloud of protein material around themselves. This is the paracentriolar material, PCM. Together, the two constitute the all-important centriole. Now, I know it gets a little confusing, especially when you're comparing. It seems like they're comparing plant with animal, But you have to understand they are in both, and they have functions in both. Okay? Function, centrioles function as a pair in most cells in animals, but as single cell centriole or basal body in flagella. Centrioles in pairs. Cells entering mitosis have a centrosome containing two pairs of centrioles and associated paracentriole. I'm sorry, I'm botching these names. Um, Material. During prophase, the centrosome divides into two parts, and a centriole pair migrates to each end or pole on the outside of the nuclear membrane or envelope. At this point, the microtubules are produced at the outer edge of the pericentriolar material and grow out into a radical or radial form. The centriole pair and PCM is called an aster, and microtubules from the aster at one pole grow towards the aster in the opposite pole. These microtubules are called spindle fibers. Some of these will become attached by centromeres or probably two chromosomes lined up on the equator of the dividing cell. Others do not, though not attached to chromosomes by centromeres, will assist in pushing apart the two parts of the dividing cell. And trying to keep this from being too complicated, I make it more complicated, but we'll continue to look at this stuff. I know it gets kind of deep. You're trying to figure it out, and we'll work on it together. We'll get this figured out. Okay? It's good to see you. Thank you very much.